Hello everyone, welcome to the Knowledge Hub by Shri Chaitanya. This is Syed Danish Anjum, Senior Zoology Faculty, and today in this session we are going to continue with the remaining basics of the classification. So in this session we will discuss about the germ layer organization, the categorization of the animals on the basis of the germ layer, and the coelom formation, and the categorization of the animal on the basis of presence or absence of coelom. So as you can see that during the embryonic development, the fertilization occurs by the fusion of male and female gamete. As a result, zygote is formed, and after the development of zygote, here we can see in the form of cleavages, in the form of mitotic divisions, when the zygote development takes place further, various cell stages are formed like 2-cell stage, 4-cell stage, 8-cell stage, and similarly morula, blastula, these all stages are forms in the embryonic development. Now as we can see that during the blastula development, it is considered as a hollow ball of mass of cells, and we can see that if we take a cross section of this blastula, we can see that in the center of it there is a cavity present which is called as blastocele. Okay, so when further development takes place in this blastula, we can see that the next stage in the form of gastrulation occurs, in which migration of the cells takes place towards the blastocele cavity. So in this migration, in this stage, germ layer formation occurs. So we can see that in the gastrula stage, we can see that there are two layers. We can see endoderm as well as ectoderm. Outermost layer the ectoderm, and the innermost layer in the invagination part is the endoderm. And during this development, as the invagination continues, we can see that there is a small pore opening called as blastopore. Now in this opening blastopore, we can further categorize whether if it is developing in mouth first or whether it is developing in anus first. So it will be either protostomiates or it will be either deuterostomate respectively, right? So this is the embryonic development which is taking place, and we will describe the formation of germ layer in this development only, and followed by coelom development, taking this development example only, right? So if we can see that during the gastrulation stage, the movement of cells occurs, morphogenetic movement takes place, and as a result, two germ layer, immediately we can see that the outermost layer, the ectoderm forms, and the endoderm is the innermost layer towards the blastocele cavity forms, right? And as a result, a small pore opening also forms called as blastopore. Now, so on the basis of how many germ layers are differentiating, how many germ layers are forming, we can categorize the organism either into diploblastic category or into triploblastic category. So if there are only two germ layers forming like ectoderm and endoderm, then the organism will be said to be diploblastic. And if all the three germ layers, all the three primary basic germ layers are forming, we can call them as triploblastic, that is ectoderm, endoderm as well as mesoderm. So here we can see in the diagram that during development in both the stages, in both the categories, ectoderm and endoderm both forms first. In the diploblastic there will be no mesoderm, but in the triploblastic, mesoderm cells will be forming only after the formation of the first two layers. So here the mesoderm will also be forming along with endoderm and ectoderm in the triploblastic animals, right? So examples we can see of the phylums, those who are triploblastic. So in the classification of the animal kingdom, the first triploblastic which occurs are the platyhelminthes, and from platyhelminthes to chordates, all are triploblastic. The examples of diploblastic are the only two phylums, Coelenterata as well as Ctenophora. So I will write it down, Ctenophora also. So these two phylums are the example of diploblastic, rest all the phylums are the example of triploblastic. Now as we see that in the diagram of NCERT also, we can see that the germ layer organization in the diploblastic as well as triploblastic, here we can see the difference that in diploblastic organism that the two germ layers are present, but in the middle of these two layers there is a third layer which is called as mesoglia. Now as we discuss about the mesoglia, mesoglia is sometimes defined as that middle layer which is non-cellular or maybe cellular in some forms, and it is usually jelly-like. So we can define it as a gelatinous layer which is present between the ectoderm and endoderm in the diploblastic organism. In this mesoglia, various other kinds of cells along with various other kinds of supporting substances can be present which are responsible for endoskeletal support. So we will see in which type of organism the role of mesoglia is what particular, right? So in diploblastic we will have mesoglia, but in triploblastic instead of mesoglia we will have a proper third germ layer which is the mesoderm. So this is the diagrammatic representation. Now on the basis of the blastopore development, we also categorized organism into the two categories. The first one is the protostomiates and the other one is the deuterostomiates. Now, how do we define the protostomiates? Protostomia and deuterostomia. That means when the blastopore development occurs in the formation of mouth first and the opposite part as the anus, then such kind of development is called as protostomiate development and the organism will be categorized as protostomiates. On the contrast, if the blastopore development if this pore is developing into anus first and the opposite pore is developing into the mouth, then such kind of organism will be termed as deuterostomate. So on the basis of the blastopore development, we can categorize either into protostomiates or either into the deuterostomiates. So here we can see that if it is forming mouth first, then it will be termed as protostomiates, or if it is developing into anus first, then we will call it as deuterostomiates. So this is the distinction between protostomiates and deuterostomiates. So the cavity which you can see during this development, this is cavity which is called as archenteron cavity. So this cavity will be developing as gut cavity. So here we can see the gut cavity, and the one opening of the gut cavity will be serving as mouth opening and the opposite end will be serving as the anus opening in the complete digestive system. Fine. So as we move further, this is the diagram from the NCERT. So this is the coelom representation, and the organism on the basis of coelom can be categorized as acoelomate. That means they are having no body cavity or no coelom cavity. Pseudocoelomate, that means they are having cavity but in the form of scattered pouches. And true coelomate cavity, that means they are having a proper coelom cavity which is enclosed by mesoderm germ layer. So we will see how the differentiation occurs. That if you see the triploblastic animals, why we are taking triploblastic? Because in diploblastic mesoderm is absent, so there will be no chance of categorizing them as coelomate or not coelomate. They will be acoelomate only. So in triploblastic also, if the third germ layer is present, still some of them can be acoelomate, for example platyhelminthes, and some could be pseudocoelomate, for example nemathelminthes, aschelminthes, nematodes, and the other can be categorized as eucoelomate. Now, as you can see that in eucoelomate, U means true. This can further be categorized into other two parts or other two categories. The first is the schizocoelomate and the other is the enterocoelomate. So if we compare between the schizocoelom and enterocoelom, we will see that it is found in the more advanced and it is found in the little primitive category of organism, but both are coelomates only. The method of development in these two are different. We will see how the schizocoelom development takes place along with the enterocoelom development. Okay, so starting with acoelomates, here we can see in the triploblastic organism, for example, this is the example of platyhelminthes flatworm planaria. If we take the cross section, we will found that outermost layer epidermis, it is of ectodermal in origin. So ectoderm germ layer is there, right? And the innermost gut cavity forming layer is the endodermal layer. So this is the endodermal layer. This is the gut cavity, alimentary canal. So we can write it down as gut cavity. And between the ectoderm and this endoderm, we can see that this body cavity is present, but this is not the cavity which you can see. It is actually filled with the mesodermal origin tissue. We termed it as mesenchymal tissue. So if I have to represent it, I will fill with this mesodermal derived cells, and this will fill all the space available, and as a result, in spite being triploblastic, they will not be having any cavity, and hence they will be categorized as acoelomate. And this mesodermal derived tissue is also called as mesenchymal tissue, space filling tissue. Sometimes in some literature or in some books it is also defined as parenchymal tissue, as the term being taken from the plants parenchyma, which is also a space filling tissue. So this cavity is actually filled with space filling tissue, which we called it as parenchymal tissue or mesenchymal tissue. So as a result, in spite of being triploblastic, in spite of having the mesodermal germ layer, as we can see these cells are derived from mesodermal germ layer. So in spite of being triploblastic, they are categorized as acoelomate because they are not having any kind of cavity. So for being a coelomate, the primary requirement is to have a cavity. The secondary requirement is then that particular cavity must be enclosed by mesoderm germ layer. So only and only if these two conditions are fulfilled, we can call that cavity as true coelom cavity. Okay. Now, talking about the pseudocoelomates, body cavity is although present, but it is not lined by the mesoderm. Instead, mesoderm is distributed as scattered pouches in the body cavity between ectoderm and endoderm, and it is having only a single phylum, that is Aschelminthes, similar to the Platyhelminthes, which is the exceptional and the only phylum example in the triploblastic acoelomate category. Here we can see the diagram that the epidermis is there, muscle layer is there, which is of mesodermal origin. So we can term it as mesodermal layer, and there is a cavity which is incomplete pseudocoelom because this particular gut cavity is having endoderm. So although the cavity is present, but it is not completely enclosed by mesodermal layer. Instead, one layer is mesodermal, but the other layer is endodermal. So it is not the true coelomic cavity. Instead, we can call it as primary body cavity or hemocoel, right? So if we have to move further, but before moving further, I would like you to scan this QR code and to get registered with us so that you can know more about the courses which are going on. Okay. Now moving further to the true coelomate category, we can see that that body cavity which is lined by the mesodermal layer or which is enclosed by the mesodermal layer is called as coelom or eucoelom. So mesodermal layer lining, ectoderm is parietal peritoneum, and that lining internal organ is visceral peritoneum. So example we can see Annelida to caudate. Now you can understand the definition in this diagram that epidermis is the outermost layer and the parietal peritoneum, that is the lining of the body cavity, inside body cavity, we call it as parietal peritoneum. So lining of the body cavity is of mesodermal origin. Retroperitoneal organs are those organs which are attached one side to the body cavity lining. So that is a different case. Now coming back to this alimentary canal, we can see that there is another peritoneal layer present outside the alimentary canal layer. It is also of mesodermal origin. This is called as visceral peritoneum. So both parietal peritoneum as well as visceral peritoneum are mesodermal in origin. So we can now say that the cavity which is being present in between these two layers is a coelom cavity, is a true coelom cavity, as both the sides are covered by mesodermal origin layer of cells. Fine. So these two are mesodermal in origin. You must remember that. Correct? So this is the true coelom cavity. Example you can see from Annelida to chordates. Now coming back to the next point, which is the development of the coelom via two different methods. One is schizocoelom, the other is the enterocoelom. Now what is the basic difference we will see in this slide. Schizocoelom is formed due to the splitting of the mesodermal layer. So where is the mesodermal layer? This is the mesodermal layer. This which I am marking, this is the mesodermal layer, or this is the mesodermal mass of cells. So in this mesodermal mass of cells, in these two layers, the splitting will take place inside and there will be a formation of a small cavity. Like you can see, when the mesodermal mass cells have developed a cavity inside them, then such cavity is appearing as a little cavity in between, a little cavity appearing in between the mesodermal mass cells layer, and further when this cavity develop and acquire whole of the space, then it results into the developing of coelom cavity. Fine. So it develops in both the ways. Both the mesodermal layer develops the cavity, and when they finally fuse at the top and the bottom part, then this results in the formation of whole body cavity, which is marked or which is lined by the mesodermal layer and hence called coelomic. Right? So this is the schizocoelom development method. Now let us take a look at the enterocoelom development method. Now enterocoelom is formed due to the outpouching of gut wall. Now what is the meaning of outpouching of gut wall? As we can see that in this method, that this is the gut lining, this is the archenteron cavity, this is the blastopore. We can see that small pouches of cells of the mesodermal origin originates in this layer only, the gut layer only, and they get separate, and in them the coelom cavity starts to develop, and further in the schizocoelom method, similar to schizocoelom method, they will completely acquire sooner or later the whole cavity and will form the true coelom cavity. So ultimately coelom is developing in both of these methods, but the starting or initiating method of developing the coelom is different, on the basis of which we have categorized them as schizocoelom if it is the mesodermal splitting method, or enterocoelom if it is outpouching of the gut layer. Right? Now moving to the next part, as we can see, the next basic phenomena is the segmentation. Now this is the term, students, which many of us get confused with true segmentation, pseudo segmentation and metamerism. So I will clearly define each of these terms here. Now what is the meaning of segmentation? That in the primary definition we can see that the body is divided not just externally but internally into segments, which we can call them, each segment we can call them as somites or metamer. The later term is more prevalent in use, with serial repetition of at least some organs ya tissue. Now there must be a serial repetition. So if all these conditions are fulfilled, then such a kind of segmentation will be called as metamerism. Right. So such kind of segmentation will be called as metamerism. So only when all the conditions are fulfilled, like has been explained in the NCERT also. It has been explained in the NCERT also that for the metamerism, the segmentation should be external, or we can put it up like that: external segmentation corresponds to internal segmentation, followed by the serial repetition of at least some organ or some structure or some tissue in each of the segment. So when all of these conditions are fulfilled, then such a segmentation only we can call it as metamerism. Right? So if I have to explain it further, we can see this is the figure of earthworm only. So if I have to draw the segment of earthworm, let us suppose I am drawing a primary schematic diagram of earthworm. This is these are the segments, right? And this is the alimentary canal, for example. By the green color I am showing the alimentary canal. Now Annelida are the coelomate animals. That means the cavity which will be present between the body wall and the alimentary canal will be the coelom cavity. I am not marking it with mesodermal layer, so we can understand. But as according to the definition, what it says of the metamerism, that external segmentation which is appearing externally in the form of circular rings in the earthworm now corresponds to internal segmentation. That means the coelom cavity will also be divided due to the internal segmentation. So here we can see that internally also the body is divided. As a result, multiple or different metamers or somites or segments which are similar, which are alike, will form due to this internal division also. Coelom cavity will also be divided, and the other condition is serial repetition of some tissue or organs. So for example, I am drawing here in the name of serial repetition. We all know that Annelida is having excretory structure nephridia. It will be explained in the upcoming phylum also. So I will draw, for example, nephridia in each segment. Then if this is the nephridium, then there will be repetition of nephridia in each particular segment, and as a result the other condition also gets fulfilled. Now nephridia is an excretory structure. It excrete out nitrogenous waste outside the body through its pore. So for example, these nephridia are distributed in each segment, so then the other serial repetition condition will also be get fulfilled, right? So this particular condition is called as metamerism. Now if we talk about the other segmentation, for example, the other segmentation could be heteronomous segmentation or homonomous segmentation. We will take a look each one by one with the definition part, but first let's take a look at how the exams are coming, or how the exam NEET questions are coming in the examination. So here's a single example. In some animal groups, the body is found divided into compartments with serial repetition of at least some organs. Now students, we have to be emphasized on the term serial repetition, because segmentation could be many ways. It could be in many ways, it could be other segmentation also, but serial repetition is also a must condition. And the other second important condition is that the adjacent segment must be in a coordination with the neighboring segment, or the segment like this segment. For example, this segment must be in coordination with its adjacent or neighboring segment. Likewise, this segment must having coordination with its adjacent or neighboring segments. For example, each of the segments are not behaving independently. They are having coordination with their adjacent neighboring segment. So this is also a very important characteristic for defining true segmentation, right? Now, so we can see that in some animals group, the body is found divided into the compartment with serial repetition of at least some organs. This characteristic feature is called as metamerism. Metagenesis is the property of cancer. Metastasis is the property of cancer. Metagenesis is the property of phylum Cnidaria, alternation of generation. We will study in the next phylum and next session. And metamorphosis is the development of larva into the adult form. So we will also study about this in the upcoming session. Now, as we move further, that segmentation can also be defined as heteronomous or homonomous. I must make a little correction. Here it is the homonomous correction. It is the homonomous segmentation. Now, if I have to define this segmentation property, that is the most primitive type of segmentation. So if the segmentation or somites or metamers of the animal are all alike, that means they are all same. Like in this diagram, I have explained that all the segments are appearing morphologically and to some extent anatomically very similar to each other. So in this type of segmentation, the segments or somites are all alike and similar. The segmentation is called as homonomous metamerism. For example, Nares, earthworm, claymworm. Fine. Now, if we come back to the other type of segmentation, that is heteronomous segmentation, we can see that in arthropods and chordates, the segments of the body are dissimilar. For example, if you look at this figure, we can see that there are major three segments we can dictate here. The first segment is the head segment or head part. The other is the middle part or the thorax segment. Now this can also be divided into subdivisions like prothorax, mesothorax, metathorax, and the rest of the part in the green color is the abdomen, and it can also be divided into further segments like abdominal segments. So in basic, as we see from the outer view, we can say that the whole body is divided into three major segments: head segment, thorax and abdomen. But these segments, head, thorax and abdomen, are all dissimilar, are all morphologically and anatomically dissimilar. So such type of segmentation we will call it as heteronomous segmentation. So example, head, thorax and abdomen are group of segments called tegmata in insects, right? So heteronomous, homonomous and metamerism. Now what is pseudometamerism or pseudo-segmentation. Pseudo-segmentation. Pseudo means false. So what is the meaning of pseudo-segmentation? As we can see in the diagram, this is the diagram of Taenia solium, a type of flatworm. Platyhelminthes phylum it belongs to. So we can see if it's body, then this is the head part, this small part is the neck part, this one, and the rest of the body is called as strobila, which is in the form of various segments. Now, if you see such type of segmentation in tapeworm, for example, new segments, proglottids, which are called as, are added from the anterior neck part, unlike earthworm. So what happens that each segment, when it progresses, it behaves independently. It is not having coordination with its adjacent segment, and at very mature stage, each segment gets detached by the process of apolysis. So this type of segmentation in which the segments are behaving more or less in an independent manner and they are not having coordination with the adjacent segment, such type of segmentation is termed as pseudo-segmentation or pseudo-metamerism. Fine. And now the last characteristic is the notochord development. So what is a notochord? So if I have to define notochord, in the simpler term I can say that it is a supportive structure which provides endoskeletal support. It is of mesodermal origin and it develops at the dorsal side below the dorsal hollow nerve cord and above the alimentary canal. So in the diagram you can see that this is the dorsal hollow nerve cord at the very top. The anterior part is forming the brain, this one, and the rest part is developing as nerve cord. Like we are having spinal cord and brain. And below this dorsal hollow nerve cord, there is a mesodermal origin tube which is termed as notochord. So it provides endoskeletal support and it is above the alimentary canal. So what are the characteristics? Again, mesodermally derived rod-like structure formed on the dorsal side. So this is the dorsal side, this is the ventral side. During embryonic development, based on notochord, animals can be categorized either into non-chordates or into chordates. So if at any stage of the life, any organism is developing notochord, whether it is persisting or whether it has been degenerated, but at any stage if the notochord appears, then such organism will be categorized into notochord or chordate, and the rest of them which are not having notochord appearance in any of the stage of the life, they will be termed as non-chordates. So there are two major categories, non-chordates and chordates, on the basis of the presence of notochord or appearance of notochord at any stage of life cycle, right? So notochord may or may not be replaced later stages in life by vertebral column. For example, we are vertebrates. We are also having notochord during our embryonic development, but later on notochords degenerate and it has been replaced by the spinal cord or the vertebral column, more appropriately, which is having the spinal cord or which is protecting the spinal cord. So if the notochord is replaced by vertebral column, they will be termed as vertebrates, and if they are not replaced, they will be called as invertebrates or non-vertebrates. And if we see the vertebrae, that means the vertebral column which is composed of vertebrae, then each vertebrae have the intervertebral disc, and the central part of that intervertebral disc has nuclei pulposi or nucleus pulposus, which is nothing but the remnant of the notochord which was formed during the embryonic development after being replaced by the vertebral column. So similar to this notochord development, there is another interesting feature which is called as cephalization. Now what is cephalization? First we will have a look at the definition part. Differentiation of head end is observed only in the bilateria. So this is a property which is concerned or related to which organism? Which are having bilateral symmetry. So bilateria. And what happens in this developmental process? That nervous system along with the sense organ gets concentrated to the anterior part of the body, somewhere near the mouth part, somewhere near the mouth opening. So concentration of the nervous tissue or nervous system along with the sense organs on the anterior end of the body, which is having mouth opening, leads to the formation of head in total, and it is the most efficient to sense and respond to the environmental stimuli, particularly finding the food or prey, right? So with the sense organ along with the nervous system being concentrated on the anterior part near the mouth, the probability of getting the food increases. As a result, cephalization is the property of forming the head towards the anterior region, right? Here we can see the concentration of sensory organs. What are sensory organs? For example, we are having sensory organs for visual like eyes, for auditory like ears, for taste like tongue, for smell like we are having nose. So these are the sensory organs. So all organs along with in the body, all the organs along with the head is focused or concentrated at the anterior part or near the mouth opening. So this is the property of cephalization. Now, here are some of the fill in the blanks for you to have a look and to give the answers. I will be explaining the answers in the next slide. First you have a look at the questions and you try to figure out the answer by yourself. So have a look. This is the questions on the basis of diploblastic and triploblastic organization. Coelom, on the basis of coelom, some questions are there. Let's move to the next slide. You can pause if you want. On the basis of segmentation, notochord. So you can have your time with these questions. These are nothing, just but a single word answers are required for these fill in the blanks. So I am now going to show the answer slide. So these are the answers, right? First of all, you try it by yourself and then you can have a look at the answers. So at the end, what are the evolutionary milestones during these developmental characters? First is the tissue grade of organization, which is the important milestone, because from this tissue organization only we are having the proper coordination, development and division of labor, and further from tissue only we are having organ and organ system level. Second is the bilateral symmetry, which is an advantageous phenomena, because as we have seen in the cephalization also that it is a property of bilateria, that is those organisms which is having bilateral symmetry. And the third category is the development of coelom. Fine. So why is the coelom is more important? Because it is the cavity which provides space to the internal organs. So the coelom development is also a advantageous phenomena and it is an important milestone in the evolution development, in the evolutionary course of time. So this was all about this basic fundamentals of animal classification. You can further, I would like to remind you, you can further scan the QR code and get registered with us. You can know more about the courses. Stay tuned with us. We can have more session and we can have more interaction. In the next session, we will continue with the phylum Porifera. Thank you so much.